My name is Sabine Seidel. I worked at the University of Bonn until last year's summer in the extant cluster Phenorock, the one or other of you may have heard of. And I took over the professorship of organic farming also this year in Vienna. And I would like to talk about optimal plant combinations and intercrops and a modeling approach, which means that I will mainly talk about experiments, but also about modeling. For the introduction, intercropping has several advantages over monoculture. This you all know, and now that Book has today found out that I'm from organic farming. Yeah. I would like to take the opportunity to say something a bit maybe out of the scope, but I think the beauty of intercropping is actually not its yield advantage. There is a yield advantage, sometimes, not always. It's often also not so big, but the beauty of intercropping is the enhancement of biodiversity. Think about the microbiome in the soil, think about insects, so product from pollinators, antidotes, and so on. And on the other side, we can use less inputs, less pesticides or no pesticides, less mineral N. And if you recall the slide that Peter has shown at the very beginning, the potential boundaries, we see that especially in these 2 points, the NNP geochemical flows and in the biodiversity, here we're having the main problems. And now I'm a bit, I have a strong opinion on that, but we have no problem, we have no lack of food in the world. If you look at the FAO homepage, they state that we have in general enough food worldwide, but it's a matter of hunger, crisis, no access to land and all of that. And I have to say that in my whole lecture, I will talk only about yield, which is maybe also, but it's a matter of a time, of course, and also the colleagues talk about it, a lot about it. But I think all the other side effects may be even more important, and we should really have them in mind. Yeah? So maybe the challenge next time, I give a yield-free lecture. Yeah. Interesting. Yeah. Maybe also for me. Okay, so what I want to say, what I want to address, or the challenge is that there is this complexity of G times E times N interactions, and then don't think I have to go into detail. We have heard that a lot. We know that a lot now. And models can support us in interpretation of data and experimental results. They can try to disentangle factors. So was it the end stress? Was it the water stress? When did the stress occur? Who was affected when? And they can also be used for generalization upscaling. And I want to give you some examples on that. Or I want to say it in other words, I mean, I have a strong background in experimentation, and these are sentences maybe all of you have heard that I've ever done field experiments, something like, what led to the yield decline? Was it now the water or the nitrogen deficiency? Actually, we don't know. Or why didn't we measure soil moisture? It would have been so helpful afterwards. Or now we have 2 years of field data. Actually, we need 3 years. We should also have not also sandy soil would have been nice, and we should have tested more cultivars. You know, these are general outcomes or general regrets you have within or after a project. And here modeling can help to some extent. So what will you learn? I will also talk a bit about field experiments, but I will not give you details. I will show you research questions that are raised. And I will tell you what process-based models are, or at least to some extent, and then I will explain you, or show you how we can use them to better interpret experimental data, to better understand interactions in intercropping. How can we use them to fill data gaps or to simulate variables we haven't measured? Variables that are hard to measure, like subsoil and uptake. And then also, which is actually the title was given to me, how we can use it also to identify maybe interesting traits or the effective traits. So this is the outline. I will say a bit about experiments, not going into detail. I will tell you what models are, and then we will focus on 2 research questions. How can we use models to interpret experimental results, and how can we use models to find under which conditions intercropping performs well? First of all, I would like to very shortly show you, present to you an experiment that maybe Thomas Döring has explained to you already. He gave a talk on, I think, Tuesday. That was in my time when I was still in the excellence cluster was on, and I was involved in the trial. So the trial was a bit similar to yours. We had talked about it. Okay, so we had a very large field experiment where we tested 4 environments. It means 2 sites. One was the conventional one, and another one was organically managed in 2 years, which are the 4 environments. And then we had 320 plots per environment. And we tested 2 genotypes, spring wheat and faba bean. We tested 2 crops, spring wheat and faba bean. And for the spring wheat, we had 10 cultivars. Yeah, 10 cultivars, and we had 2 faba bean cultivars. And then we tested also 2 sowing densities, and we grew them as intercropping and the soil crops, of course. And then we had a strong hypothesis at that time. I have to say that it was a full mixture. You can see here the images where you see we grew them in rows with 21 centimeters of distance, and they were fully mixed. And the hypothesis was that the spring height affects the mixture performance. So the taller the spring, the more suppressed the faba bean. That was our hypothesis, and this is why we choose 10 cultivars of spring wheat with ideally, which never works out, as you know, decreasing height. And the 2 faba beans, faba bean cultivars, also one was supposed to be a bit taller than the other one, which was in the end also not so super. The difference was not so much. And now you see this very super strong correlation, which is the total yield in the mixture over the spring wheat height for the 2 faba beans. And you can see that, well, if you're very optimistic, you see a bit of a correlation. And this was only in 1 out of the other environments where we found it. So in the other ones, we found no correlation even. So the spring wheat height had really a minor effect on the performance. And so what are the key insights? As I said, I cannot go into detail. I will tell you the key insights, and you can look them up later or ask me later. What is very important, which I didn't believe really for a long time, and now I really have to say it's like that. The species interact at a very early stage, even very shortly after emergence. And this has been also shown by Thomas Döring in other publications and other experiments. So very early, they already interact, and then you have a very strong legacy effect. So you have a very strong, for instance, a very early cereal that is very strong at the beginning, very dominating, it keeps on being dominated, generally said, of course. Yeah? And then, of course, the mixture effect depends on sowing density, the environment, and the combination of the genotype and the 2 genotypes. But we could not really find out. We could not really find out what actually was really key. And one of the outcomes is really that it's very complex to analyze these experiments. And even with all this effort we did in this experiment, you can imagine how much work it was. It was really difficult to draw out general conclusions. So which crop traits are key? It wasn't the plant height. And what are the effects of management and environment? And this is another example. It is a following up experiment where we selected only 2 of the spring wheat cultivars. And I don't want to go into detail now, but it's the same site, very same setup. And the land equivalent ratio, I think I don't have to explain to you, right? Does anybody not know it? Okay. So this is the land equivalent ratio of 1 year. And this is it for the second year. And here we have in brown the spring wheat cultivar Annabelle, and this is the spring wheat cultivar Ahab. And in 1 year, which was a very dry year, we had a very strong cultivar effect. These are 3 N levels, but this we ignore now. And in the other year, it was not the case. So even some years you have a genotype effect, and then in the other you don't have. That makes it even more complicated. Okay, so, and I want to show you these pictures to illustrate also how very differently the plants look like. So that was a very dry year. You can see here, there are very few bean plants. You can hardly see them. You can also see that the spring week is really short and really suffering from drought stress. And this is then the second year where the Faber bean was even taller than me. Hard to imagine. I could even hide in it. And you can see here, hardly here the spring wheat. So it's really fully different story, just because of the water or the precipitation. And then I told you also that we did some root nightmare and exploratory work. We took a course up to 1 meter depth with this electric camera. And we discriminated between the species with FTS spectroscopy, and there we tested also the effect of different sowing densities, which was very strong, which is much stronger than the cultivar effect. And the lower sowing density especially fostered deeper rooting, whereas when we had a very high sowing density, the plants invested a lot in the shoots. They try to get the light, and they invested much less into the roots. And then we also identified root traits that were beneficial in intercropping. And the major root trait is actually the early vigor. So the early growth of the roots really play a major role, especially the early growth of the dominating cereal. So if you have a dominating cereal that grows in the beginning, also develops a lot of roots, the chance is quite high that it will suppress the Faber bean.
When we're talking about early vigor, it's for both species at the same time, or it's more on one species or here?
So we sampled at the same time both of the crops. Yeah.
So maybe we can play on that
with the
time to be to guide the.
Yeah, theoretically yes. Practically, of course, you cannot. You have to sow them more or less at the same time. At least when we talk about spring crops, right? I mean, that's not so much time.
When fall or that kind of thing, you can enter in the field of the world.
Yeah. Yeah, or if the one already germinated, it's not a good idea to sow them into it. Okay, so now with this, giving you the idea how complex it is, but I think this idea you already had before. But this is also, I gave the information to you because this data was also used intensively in the modeling exercise, because we always use the observations for our modeling exercises. But first, before we come to the application, I would like to give you an insight about process-based crop models, what they are. And so here's the sentences may describe them quite well. Process-based crop simulation models are software tools, dynamic tools that usually work on a daily basis, and they simulate crop development and growth in relation to the environment, which means the weather and the soil, and the management practices, which is mainly sowing date and fertilization of macronutrients based on agronomic principles. Into this last part of the sentence, I will come in the next slide. So this is just a scheme, what you need for such a model. And just maybe for some examples, you may know the model EPSIM and DSAT and CERA. So these are the models I'm talking about. Yeah? So they are usually one-dimensional, and they are used, for instance, for climate change impact studies. If you know these papers on what is the impact of climate change and wheat growth in 2100, so often these kinds of models are used. But they can be also used to provide explanations or for yield gap analysis if you compare potential with actual growth, or for decision making. There's also some decision tools. And what you need for such a model is you need model input, which is weather data of a specific site. You need soil properties of the specific site. You need cultivar properties for the for the specific crop and cultivar, and you need the information on the management. So the model is always representing, it's always a virtual field. Then it has incorporate processes. For instance, it simulates the phenological development based on the day length, the temperature, the light. Then it simulates the biomass and as a function of light interception, but I will show this a bit later. It simulates the root distribution, evapotranspiration, soil water dynamics over depth, and soil nutrient dynamics over depth. It depends a bit on the model. Some are more strong in this or more weak in that, but these are the general processes that are simulated. And it simulates all these processes that are provided as mathematical equations. So if we have knowledge about a relationship, we can implement it in the model, and then it can simulate it. If we do not know the relationship, or if we do not enter it into the model, it also cannot simulate it. Yeah? And then it provides outputs, which are usually daily, like the development stage, biomass growth, and leaching, or greenhouse gas emissions, which are two variables that are hard to measure, or at least hard to measure in a daily time step, or plant and nutrient uptake, and many others. Coming back to the economic principles, so as I have already said, there are equations in the model, and usually they are mathematically not very challenging, so they are just relationships, like here the net photosynthesis is a relationship of irradiance for C3 and C4 plants, or the maximum gross photosynthesis related to temperature, again for C3 and C4 plants, but also other principles of equations are in the models. For instance, the PF curve, the soil water retention curve, the relationship of soil water potential and soil water content, or other principles from soil science like soil organic matter, soil organic carbon turnover, soil organic carbon pools, or whatever. Yeah. And so I'm stressing this because this is very simply said, in contrast to machine learning, where you have no process in it. Yeah? So here the idea is to have a process in it. Without, I don't want to comment, I mean, I don't want to weight that or judge that, I'm just saying that. Yeah? And so one example, for instance, how is the daily biomass increment simulated of a crop? So the model calculates, for instance, the potential daily biomass accumulation in gram per square meter, has a function of the crop-specific light use efficiency. So crops have different, depending on the crop, it has different light use efficiency, times the power int, which is the intercepted photosynthetic active radiation. And this here, this power int is, again, depends on the incoming radiation, the weather data, plus there's crop-specific extinction coefficient, and the actual LAI. And the LAI behavior index, again, for instance, depends on the crop and on the N level and so on. So there are a lot of interactions which maybe people can't solve themselves in the brain, but also mathematically not super challenging. Yeah. Then here at that step, we are at the potential growth, but then the models also consider stress or limitations, which is usually drought stress, nutrient deficiency, where most models only consider N, some consider P, almost none considers K, and temperature stress, cold and heat stress. And then you have a dominant stress, so maybe in one day the drought stress is more dominant than N stress, and then this stress reduces the daily increase of that specific day by, I don't know, maybe 20%. And then the biomass that has been produced and is then allocated to the plant organs. And here we have so-called tables. So at an early growth stage, most of the carbon goes into the roots, and then at a later growth stage, most of it gets into the grains and so on. So this is then in specific tables that change the allocation depending on the development stage. And maybe, so this is a bit the same story, but from a different angle. So you have here the model with the upper boundary, which is the weather, and the lower boundary, which is the soil. And then what you need always is the weather data. What you need is the crop management, like irrigation amount and dates, fertilization, crop rotation, sowing date, and so on. You need the information from the soil. And then you need information on the crop parameters. And here, all models provide default parameters. For instance, there's a set of parameters that define Central European winter wheat in general. And then you have to adapt it to your specific cultivar by changing some of the parameters, which we call then calibration. So this means the model incorporates or considers G, which is the crop parameters, E, which is the weather, E, which is the soil, and M, which is the management.
So does it need weather information on field scale, or is the general weather station close by, kilometers away also?
Yeah, I mean, so it has to be representative, and it has to be available.
Yes,
So
available.
yeah, exactly. So experimental stations often have their own weather data. If you don't have it, you use the one that is close by and also representative. Yeah, if it's on a hill but close by, it doesn't help you. Also. Okay, then I want to say a few words about the process-based model we used, which is in detail also presented in that paper. So the model that we use is simplification. So models are always simplification, right? Whatever also the model the colleagues showed, and the same is true here. So a lot of things are not considered. It's a simplification with a focus on the key processes. So what the model here considers is the growth of both species. It considers the soil water dynamics. It considers the N dynamics in the soil, including the biological N fixation of the vacuum, and also the N uptake. And it is able to consider the competition for radiation. And here, important is the difference of the height between the two crops, but also the form of the crops, which is simplified by giving it a bit different shape. Yeah. So it's not so detailed as the FSP model, yeah, but it has different purpose. So the idea is actually you can run one simulation in 1 second. Yeah. For the FSPM, it really takes, well, I don't know, 15 minutes or whatever. Yeah. Yeah, it takes. There is this joke that it takes as long as the plant to grow. I think it's just not not that difficult, but yeah. So yeah, but it has, but it's more detailed. Yeah. So everything has its niche or its application, and here it's definitely less. It's rougher, no doubt.
Are you talking more about like spot focus type?
Yes. Okay. Yeah. Exactly. Yeah. Then complementarity. So which is the reduced niche, overlap, and compensation between the crops, but only abiotic resources. Actually, the model only considers water and competition or complementarity. So complementarity, if one of the crops grows much deeper than the other one, for instance, that can be considered. What it does not consider is weeds. And there's, I think, I only know one model from France which does that. So it's really a niche. And pests and diseases, and also not facilitation in the sense of providing P or lowering the pH value or something. Yeah? So this is ignored, but it can consider N and water, compensation and complementarity, which is, of course, plays a major role in our systems. So this is, okay, sorry, this is strange animation. I want to show you here shortly how we have calibrated and validated the model. I know this figure is a bit small, but again, if you're really interested in the source of the literature I have shared to you beforehand, you can look it up there. So what we have done, we used the data from 3 of the 4 environments. And we used, so what you usually do is you calibrate the model against the soil treatments. That means you use all the data you have, the weather data, soil data, and so on. And then you run the model against the observations. And if there is, for instance, the absolute yield is a bit lower. You increase, for instance, the radiation use efficiency, or you adapt a bit the model. And then you validate it against the intercrop treatments. And you can see that here, this is the grain yield for 3 environments. This is the fava bean intercropped, and this is the spring wheat intercropped, and you can see, so more or less the model performs okay. Yeah, so it's more or less the same. So sorry, this blue is the simulation, and green is observation, and we simulated various cultivars. This is why it's a box plot. Yeah, and of course the observed box plot is larger because you have also measurement errors or something that you don't capture with the model. But the model fitted quite well. Here we have a more discrepancy, which is can be explained by the fact that it was the organic side, and we had here high wheat pressure, which led to an overestimation by the model because the model doesn't consider the wheat, right? So this is kind of the effect, the loss due to the wheat can be also interpreted like that. And we looked at the results from different angles. Here, for instance, we also looked at the LER and compared it to the observed versus the observed versus the simulated. Green is the PLER of fava bean, and blue is the PLER of spring wheat. And these are the 3 sites. And you can see that the model was able to simulate the increased LERs quite well. Okay, now we're coming to the third part. Can models help to interpret experimental results and disentangle complex interactions? First of all, we used then the model to simulate water uptake. And you can imagine it's quite difficult to measure water uptake, and especially to measure water uptake for specific soil layers. You need isotopes, or it's really a super complicated thing. So this is really a task, or a good task for a model. And sorry, I forgot to say before, when I showed you the simulation and the calibration results, that we have not only looked at yield, but we have also done a lot of measurements in between the season, like LER and biomass and height, and all of that was also considered in the calibration. Yeah, I just showed you now the yield result to not overwhelm you with figures. So we had also some measurements of soil moisture, which confirmed. We had measurements of soil moisture in different depths, which cannot confirm water uptake, but it confirmed the simulated soil moisture. Yeah? And what you can see here, it's a bit difficult, but once you understand it, it's not so difficult anymore. So it's the water uptake in millimeters, and this bar here accumulated over 1 season. So it's just for one of the seasons. And this bar here is the average of the monocrops, of the 2 monocrops, and this is the value, the simulated value for the mixture. Yeah, and then we have the same value here, the monocrop, the spring wheat uptake, and this is the spring wheat monocrops. No, this is the spring wheat in the mixture. This is the fava bean monocrop, and this is the fava bean in the mixture. And what you can see here, if you look at the first 2 bars, you can see that the water uptake in the mixture versus the monocrop is actually the same. Yeah? And this is also what we found in our measurements of soil moisture. But measurements of soil moisture are always very heterogeneous, and it's not so easy, but in general, we did not find any significant differences. And so the water uptake was quite similar, but what was different is actually the share. So what you can see here is that the spring wheat in the mixture takes up much more water as compared to the monocrop in the deeper soil layers, yeah? Which is then actually at the expense of the fava bean, because the water is then of course gone, and the fava bean can take up less water. Another example is simulating values that are hard to measure is the N uptake. So here we simulated the crop N uptake plus the biological fixation of the fava bean. It is the same figure. Again, here, the average of the monocrop, the average of the mix, then here spring wheat and fava bean, and the colors are again the soil depth. Sorry, I forgot to mention it before. I hope you saw it. So this is the top layer from 0 to 30, and then 30 to 60, 60 to 90, 90 to 120. And the dark bluish is the end fixation. And what we found here is that the intercropping reduced the absolute end fixation by 25%. Do you have an idea why it is the case? Why do we have a decline of absolute end fixation in the intercrop?
What's too much competition for phosphorus? So
the phosphorus is not limited here.
Bees prefer to get the nutrient from soil before fixation. Maybe as the weeds compete with them only, they try first to get the nutrient from the soil, and they didn't make it for a while. So after a while, they have less resources to make fixation.
Yeah, so that is one of the reasons. And the other reason is that the fava bean in general was suppressed very strongly. So we had a lower proportion of fava bean. Yeah? So these two reasons. And then also, if you have, of course, less fava bean, they can take up more from the soil. Sorry.
I don't understand. Actually, I don't understand the figure very well. So blue is total N up fixation.
This blue. This is the N fixation.
Oh yeah, it's the portion of N fixation of total N uptake, and the rest is the uptake from the
soil. Exactly. Yeah. Okay. Is that clear? Sorry if I'm too fast. Just.
It is clear. I was wondering if they have the same nitrogen application in both situations.
So the spring wheat received fertilization. Don't name it down. I think it was maybe something like 50 kilograms, not too much. Yeah? And then the intercrop, it was half of it. And the fava bean didn't get any.
Okay.
So this could also explain the effect.
Yeah.
The fact that it was fertilized in the mixtures means that indeed.
Yeah. There was enough,
Could have been
and
easiest.
there was enough, and there was less need to take the atmospheric one. Yeah, of course. But what we also see is that the spring wheat N uptake from the soil has been increased. So this is a mean. It's not exactly the value you see here, but it's a mean over all environments. What you can see here that spring wheat mix, the N uptake in the mixture, especially in the lower layers, is higher as compared to the monocrop, which is also clear because it has just more nitrogen away. Right? Okay. And okay, so what we have seen so far, we have seen that the early wheat rooting dominance definitely negatively impacted the fava bean. I have said that before, so the early growth really matters, and here especially if you have a dominant cereal, you have a legacy effect, and this we have proved in our observations. But of course, you will never have daily root observations in the field. If you do coring, we had a few dates, not too much. I would just show you in a bit a figure that from the simulations that proves this outcome. And then also the competition dominated root growth in the deeper soil layers. So especially in the deeper soil layers, the crops competed for resources. And then also especially critical phase for the fava bean was the early growth stage and the one for flowering. But actually, I have the animation was wrong. I should have shown the figure before. So what you can see here is for one environment, the day after sowing on the x-axis, and this is a factor, a transpiration reduction factor, stress factor, let's say. Yeah? And when it's 1, it means there is no stress, no drought stress, and when it's 0, there's super strong drought stress and no biomass growth takes place in the model. And what we compare here in green is this factor for the mixed fava bean, and in red it's for the sole fava bean. And there are two phases where there's a strong difference, which is especially here at the beginning. So at the very early beginning in the mixture, the fava bean experienced stress. And this is, as I have said before, also this early dominance that is negative and then has this legacy effect. And then also around flowering, again, there was a phase where drought has occurred and really affected the fava bean. So the critical phase for the fava bean in our case was the early growth stage and then before and around flowering. So in this kind of figures, the simulations really help you not only to identify which resource may lead to competence, but also when, which is of course also very, very relevant. And now I want to show you this figure. I don't know, have you ever seen this C4 approach from Eric Schiss? Have you seen this kind of graph before? This box with the, yeah? Has anyone seen it before? Yeah, okay. So it shows the P-LER of one crop. Here in this case, it's the fava bean over the P-LER of spring wheat. Yeah. And then we have 4 blocks, so 4 colors. And so it's actually just a good way for illustrating that if we are here in that A block, in this bluish block, the P-LERs of both crops are below 0.5. So the lose-lose situation. Yeah. Competition is stronger than complementarity and cooperation. This is, of course, not what we want at all. Whereas on the other side, this is where we're aiming for. The P-LERs of both crops are above 0.5, which means that complementarity and cooperation are stronger than competition. And then we have the 2 other cases. In this case, the greenish one, the cereal suppresses the legume, so the cereal has the higher P-LER and the fava bean the lower, and then the other way, the legume suppresses the cereal. And here I've plotted the simulated and observed values, and the simulated values are the ones with the black border or black border, and red is observed, and then the colors are the different sowing densities, and then you have the different environments. One environment where we have not too much and not too much water, one where we have a lot of water and a lot of nitrogen, and then this is the organic side where we have not so much. But it's not, I don't want to go into detail here now. What I want to show you is that the simulations and the observations they are not always perfectly like on each other, but the story or the message is the same, which means, for instance, here, observed and simulated are in this box, in this part, or here for the organic farming. And this is the other block, which is this conventional side in the drier year. And here you may remember before, here we have this discrepancy, which is the organic side where the model is not able to simulate the feed. And this is now the grain yield that we can have. But the question is actually for which resource they are competing for, and you can have a similar figure also for different resources based on the simulated data. Now we don't have observations here. We have only the simulated data, where you can see here for the water uptake, crop water uptake, we are all in this area of cereal suppresses the legume. Whereas if we look at N uptake plus N fixation, the picture is a bit different. Here in this site, which is a conventional site with quite some nitrogen, we are here in the cereal suppresses legume. Here we are between the 2 of them, and here in the organic managed site, which where the N level is of course lower, the complementary and the cooperation is stronger than the competition. So also, of course, the N level at the beginning plays a major role. Okay, now I'm coming to my last part. How can we use models to find where and under which conditions intercropping performs well? This is a scenario analysis, which really it's not necessary that we go into details or that you just understand how every point. What I just want to tell you, actually, my point is more to tell you how we can use this tool of the models. And what we did here, we had the model set up for one site, and we generated a weather data that is based on. So we have like 20 years of weather data from a weather station, and we put it in a so-called weather generator that just blows up the number of years. You can have then, I don't know, thousands of years, and they have all the same statistical characteristics. And we did that to have more weather scenarios. This is why you have the box plots. And you can see here the LER, and these are the different PLERs. Read the legend. You can see the PLERs. So this is the PLER of the wheat in green, and then the red is of the bean. And I just want to show you that you have different combinations of rooting depth. So here, for instance, the wheat rooting depth, maximum rooting depth was 84 centimeters, and the fava bean 78. And then the next scenario, the wheat was again 84 and 97 centimeters for the bean. So we don't have to go into detail. What I want to show you is that you can play and can test very different combinations and see and try to find a pattern in the combinations. Yeah? And this is what we have done here now for just one site, to explore the potential of the model and to get hands on it. But the task of, or the work of my current postdoc is to do this on a larger scale, which is the federal state of North Rhine-Westphalia in Germany, which is a federal state here, and Bonn is one of the cities there. And so what he used this spring wheat fava bean model he has set up for the experiment that you know already. So you remember it was this very strong mixture of the two crops, and he tested different plant traits. So we have, as I said before, we have played around a bit with which trait could be interesting, which crop parameter also can be related to a trait that we really can measure, which is also a bit tricky. And we identified 3 traits of interest. First of all, the plant height, where, of course, our expectation was already that the effect is not very strong, because this is what we saw in the experiment. And the daily rooting rate, which then, of course, determines the maximum rooting depth. And the canopy extinction factor that has to do with the light interception. And then, so we have these 3 traits, and we had different levels of the traits per crop. And we tested different crop management, also changed the crop management, or played around with the crop management, so we have different N levels also. And then we did this all for soil cropping. So we had one plant height, one daily rooting rate, and then we changed the canopy extinction coefficient for different different values, and this under one N level. And then we changed, so everything was kept stable, but just one parameter, one value was changed, which in the end leads to quite a lot of combinations, 16,000 actually, for all those that calculated it fastly in the brain. Then we used the soil data that was available from the soil inventory, which is, we had soil data from 110 sites because of the soil inventory. And we used historic data, 30 years of historic data, and this led us then to more than 3 million data points to analyze. And we did not only calculate the LER, but also the net effect, which is the yield advantage, but it's not a ratio. It has the same unit as the variable that you use. So if you look at grain yield, it comes also, or the unit of the net effect is also tons per hectare. So for instance, if you have the if you look at this, this is the formula. So the net effect for the yield, for instance, is here the yield of the spring wheat and intercropping minus 0.5 the yield in soil cropping. And the 0.5, why the 0.5? Because we have to reduce the sowing density by 50% from sow to intercropping, right? This is the factor of the reduction of the plant number. And then the same plus the same for the fava bean. Yeah, so you can come out, the result is then an increase of 0.5 tons per hectare, whatever the net effect. And I will show you some slides. I know that it's a bit difficult to show such slides really fancy, but I mean, you have only 10 minutes, so you will survive. First of all, I want to show you this map here, which shows the land use efficiency of intercrops at different sites of 210 sites, each of these plots. And the difference between this plot or panel and this panel is the N input. So here we have no N input. Here we have 20 kilogram of N input. Here we have 40, and here we have 60. Yeah, and you do not have to now find the differences because it's very difficult, of course, to see it now with the colors and so on. But here you see the mean values. So the mean LER in case of no fertilization is 1.18. And then with increasing fertilization, it's 1.16, 1.11, 1.9. So what we also proved with the model is that, so intercropping, first of all, the mean LER was above 1, was around 18%, which is also what we know from the literature. And then the net, and then we see this decline with increasing in fertilization, which is also, of course, what we would expect, right? What we also found, this is not shown here, but the figure would look quite similar, is that the net effect increases with N fertilization. So the more N you fertilize, especially the higher the wheat yield gets, and the higher the absolute yield is. But I will show a bit more on that later. So we found in general, we found a trend towards higher absolute grain yields with higher N inputs, but accompanied by a reduction of the LER. And now this is what I meant, which is maybe a bit dry, but it's really hard to show all these data points in a fancy way. But you will make it, I will guide you through. And this is important that you understand this figure because I have, I think, 2 or 3 of them. So what it shows here is the spring wheat grain yield here in this upper part, and here it's the fava bean grain yield, depending on the difference of the root elongation. The difference of the root elongation of wheat minus fava bean, which means whenever we are here in the purple side, the bean is rooting deeper, and whenever we are here in the orange part, the wheat is rooting deeper, right? So this is all you have to know. And then we have it for the 4N levels. Yeah? And what you can see is that the yield per species is strongly influenced by the root elongation rate. Yeah? So you can see the deeper the wheat is rooting, the higher the yield of the wheat, whereas it's vice versa for the fava bean. The deeper the wheat is rooting, the lower the yield of the fava bean is. And then you can also see that there is an impact of the N on all of that, but the directions are quite similar. And now let's look at the net effect. The net effect in tons per hectare for the 4 different N levels, and again the differences in the daily root elongation. So here, okay. So here, what we see here is that we have an enhancement of here. This is the net effect is here 0.5 tons per hectare. So the highest net effect, the highest absolute grain advantage we had if we have a high N fertilization and if the spring wheat is deep rooting. So there is this interaction between management and genotype. And we can also, again, show that in this figure that I have shown before, in this C4 approach, you can see here different ones. You have here the net effect of faba bean, and this is the net effect, sorry, it's the net effect of faba bean, and then the net effect of spring wheat. And we have here different differences of the root elongation. So in general, we can say that here, the deeper the faba bean is rooting deeper, and then they're getting closer, closer, closer. And here the wheat is rooting deeper. And if you just look at the two extremes, you can see that especially here we have a deeper rooting wheat. We have here the highest net effect. So the net effect is positively related with the root elongation rate and the end input. But you can also see, or what, especially in the figure before, we saw that the effect was not so super high. So it was the maximum actually were these 0.5 tons per hectare in a mean. Yeah, so it didn't really boost productivity, the intercropping. And this is my last slide in this to torture you with, or figure in that way. I know it's really a dry material. And so we did this also for plant height, and I don't want to go too much into detail. So we have again here the difference in maximum plant height, the spring wheat grain yield, the faba bean grain yield, and here on the left side purple is the tall faba bean, and on the right side is in greenish and orange the tall wheat. And what you can see is the taller the wheat, the taller the wheat, the higher the spring wheat, and the taller the wheat, the lower the faba bean yield. Yeah? But the effect again is not very high. This is also what we saw in the experiment. You can have some effects of the wheat, of the heights, but you will not explode yield. Yeah, with having the right, having a specific growth. Oh, okay, now this is really the last one. I hope so. Yeah, this is really the last one. So because we also talked about the E, which is of course E is not only weather but also soil. So we also try to find out what the effect of the environment is, or the soil is. And here you can see the soil sand texture content increasing. So it's here for sites with a top soil sand content below 27%. Up to 75%, and then again for the different N fertilization inputs, and again for different root elongation differences. So here we have in purple deep rooting faba bean and yellow deep rooting wheat. And what you can see that the net effects are generally greater on soils with higher soil content. So here we are reaching higher values. Here we are reaching higher values than here. So higher soil content, higher net effects, mean net effects can be found, but again, also the effect is not too high. So the variation is between 0.25 to 0.5 tons per hectare. With this, I'm at the end, and I want to give you a wrap up of the findings. So first of all, as I said, it was really interesting for me, and something I really didn't expect. That the early growth really has a very high impact on the overall growth along the whole growing season, and especially if the wheat is dominant, this can really strongly negatively impact the faba bean. Then the competition dominated root growth and resource uptake, especially in the deeper soil layers. It was not so much the case in the upper soil layers, but the deeper soils, who reaches the water, the nutrients in the soil layer below 60, 90 centimeters. And then, I mean, maybe this is not so super unexpected, The critical phase for the fava bean, especially the intercrop, is the early growth stage, and of course, especially the period before flowering or around flowering. For the fava bean, the water was limited. This is key to determine. This is also why I commented when you asked, because for us really the seabeds were super grew very bad if it was very dry. This is why I commented on your slide that I expected maybe that it was a dry condition. Whereas for the wheat, it was the nitrogen. So the wheat was mainly able to take up the water that was needed. The nitrogen was more of a limitation. And then the interactions between N input and potential root elongation rate had a moderate influence on the overall grain yield, but strongly affected the competition and the yield partitioning between the plants, as it has also been shown before, that maybe even if the overall didn't change so much the partitioning. So in 1 year, or depending on the trade or depending on the management, you can change a lot the proportions of the plants. So if you have just 1/3 of legume or half of or half of is legume or whatever, so you can impact more the proportion than boost the overall yield, especially in a system like we have, which is where the plants are more or less a similar height. And we have a very intensive system also, so it could be a bit different if you have a more low input system. But for this quite high input system and with this very intense growth together, it's really a different. It's like that. Also, for instance, if you have a relay intercropping system, you could gain more with that. So if you have a relay, more time difference, maybe you can even get higher yields. But here we seem to be, I mean, there is some room, but it seems to be not that we can boost the yields with the scan. Okay, with this I'm at the end, and I'm happy for questions you have.